Good afternoon. It is my great pleasure and delight to introduce today's Wednesday afternoon lecture speaker, Dr. Xuetao, the president of the Chinese Academy of Medical Sciences and of the neighboring Peking Union Medical College in Beijing, China. Dr. Cao is one of the top immunologists in China who has had a spectacular career as both a scientist and a scientific administrator. At age 28, he became the youngest person to earn the title of medical professor in China, and by age 40, he was named vice president of the prestigious Second Military Medical University in Shanghai, where he earned both his MD and PhD. Dr. Cao's laboratory focuses on the understanding of innate signaling in immunity and inflammation, the identification of cell subsets, and new molecules in dendritic cell-initiated immune response, and cancer immunotherapy, a subject of considerable importance and topicality. His group has identified important mediators and regulators of innate signaling and characterized immune subsets with regulatory function in immunity, inflammation, and cancer, which you'll hear about today. As corresponding author, he's published more than 220 original papers in numerous top-tier journals. He serves on the editorial or advisory board of a number of journals, including Cell, Annual Reviews of Immunology, and Science Translational Medicine. As a scientific administrator, Dr. Cao has been tasked to restructure the Chinese Academy of Medical Sciences, an organization perhaps most akin to the NIH Intramural Research Program, in that it includes both research laboratories and hospitals, and it really serves as one of the major focuses of academic research in China. This daunting task, he has said, involves not simply copying a Western model, but rather creating something novel that can work at a Chinese federal level and enable the youngest generation of scientists, both basic and clinical, to flourish in their home country while embracing international collaborations. Dr. Cao's many honors include the Nature Awards in Mentorship for Science, membership in the American Association of Immunologists, membership in the Chinese Academy of Engineering, the German Academy of Sciences, and the French Academy of Medicine. Dr. Cao is widely recognized as a thought leader in promoting innovative and cross-disciplinary research and has worked tirelessly to encourage international collaborations and to enhance the global visibility of China's own research and development. We are really thrilled to have him speaking with us today. Please join me in welcoming Dr. Cao.
Thank you, Michael, for the kind introduction. Good afternoon, ladies and gentlemen. It's a great honor to speak here. First, I want to thank the kind invitation to give me the chance to share the data from our laboratory in the investigation of cancer and immunity. I attended the memorial symposium for Bill Paul the day before yesterday. I attended the Laurie Grimshaw's lecture, which reminded me a lot of memorable things I had with Bill when he visited China. I want to take this opportunity to express my sincere thanks for his generous support to Immunological Society and to me in the last decades. Yeah. And also I have so many very close friends in NIH, so I would like to take this opportunity to thank you for your help also in my career. Today I want to talk about the innate molecule in the inflammation and cancer. As we know, the immunity and the cancer become more popular in the last 2 decades. And also the immunotherapy has been successfully applied in clinic. For example, the checkpoint blockade and the CAR T cell adoptive transfer. However, the clinical outcome is still limited. So how to improve the therapeutic efficacy and how to figure out what's the population of the cancer patient responsive to the cancer immunotherapy are the challenging issue to our immunologists, which need us to better understand what's the interactions of tumor and host, what's happening in vivo. So maybe there are so many mechanisms behind it to affect the efficacy of the immune therapy and also help the cancer cell escape of immunological attack. So there are lots of things we should further investigate. As we know, the fundamental function of the immune system is to discriminate the self and dangerous non-self. As we know, the immune cells are responsive to the evading pathogens, so they can be activated efficiently after they sense the evading pathogen. In the last 2 decades, we pay much attention how to activate the immune response against cancer or infectious disease. So we have identified many kinds of activators or effectors of immune system. Finally, we understand that the immune system can develop the negative regulation strategy to control the activation of their systems by upregulating the negative regulator or immune suppressive cells in vivo. So how to activate the immune response? Not only we use the activator, but also we can find the way to suppressing the suppressor. So the checkpoint blockade is the typical approach for the suppressing the suppressor, finally leading to the efficient activation of the immune response. So this is the balanced network of the immune system. How we can get the functional clues or indications from the basic knowledge is also the good question we should answer. Antigen-presenting cells play an important role in the initiation of immune response, but also are critical in the maintenance of the immune homeostasis. So how they can efficiently initiate the innate response against the infectious pathogens, because they express many kinds of the PRR, pattern recognition receptor, on their surface, which can distinguish the self and the non-self PAMs. So once recognition of the evading pathogens, the downstream pathways of the PRR can be activated. So leading to the production of the chemokines, cytokines, and also upregulation of co-stimulated molecule, or MHC, on their surface, which can interact with the T cells and B cells, so forming the immunological network to eliminate the evading pathogens. So what's the signals can be efficiently initiated, and how this signal pathway can be timely determined at the appropriate level to avoid the overactivation and prevent the harmful immune pathology is also a good question we immunologists should answer. So this, in the last 2 decades, my laboratory focused on the immune biology of the antigen-presenting cells, including dendritic cells, macrophages, and investigated how they can be activated and what's the signal pathways inside to be regulated at the cellular level and molecular level, and what's the immune response, including the innate inflammatory response, can be initiated. And also, we are very curious how the inflammation can be resolved. So we investigated the cellular and molecular mechanisms for resolving inflammation, especially at the antigen-presenting cells level. And also we're very interested in the interaction of the cancer cell and the immune system. How the immune cell can escape the immunological attack by upregulating the negative regulators or by reducing the activation of the immune response. And also, how about the immune system educated by the primary tumors to create the favorable environment for circulating tumor cell colonization or for the future metastasis in the distinct organ? We're also very interested in this. Also, we try first to translate the basic knowledge to the clinic, trying to set up the new approach to the cancer immunotherapy or identifying the biomarkers to predict the prognosis of the cancer patients, especially receiving the certain kind of the cancer immunotherapy. So why we ask the question today, I would like to share the data about the role of the Innate sensor, actually two important RNA sensors, TLR-3 and RIG-I, in cancer progression and immunotherapy. I have several slides to briefly introduce the background, why we ask this question. So this, now there is a well accepted concept in the tumor immunology is immunogenic chemotherapy. The chemical drugs can increase the immunogenicity of the tumor cell and also release the alarmins from the cancer cell to activate the immune response. This is the new concept now. 20 years ago, this is, I had the first original paper in English published in Western countries. That's about which describes the combinative use of alpha interferon gene therapy with adaptive chemoimmunotherapy. So it's also, we investigated how the therapeutic efficacy in the model, in the tumor model, bearing a human hepatocellular carcinoma. It is, has the highest incidence of mortality in China. So this study inspired me to think about what the chemical drugs can modulate the immunogenicity of cancer cell. So, and is it possible for me to find the new activating molecule or adjuvant molecule to activate dendritic cell or other cells which can be used for the adaptive transfer or for the vaccination? So we contracted human dendritic cell seeding library and did a large-scale automatic screening at that time, identified several novel molecules, including one, which is the HSP70L1. So we started to investigate the function of this molecule on the dendritic cell activation and antigen-presenting capacity. Finally, we find this is the new adjuvant. It combined to the TLR-4 receptor on the surface of the dendritic cell and activates or increase the antigen-presenting capacity of dendritic cell to activate T cell response, finally killing the tumor cell. And then we think about how about the heat shock treated of the tumor cell. Then we analyze the immunogenicity of the tumor cell after heat shock. And also we find they can release the heat shock proteins and also have high expression of heat shock protein intracellularly. So we find this is the good adjuvant. And also heat shock protein can increase the expression of the MHC class 1, class 2. So having the increased HSP antigenic or immunogenic complex at that time. So we also use this complex to parse the dendritic cells and use the dendritic cells to vaccinate the tumor-bearing mouse. We found the increased CTL response can be achieved. So this reminds us we can find the ways or identify the adjuvant to activate anti-tumor immune response. Yeah. So this is the good. Then we started to do clinical trial. This clinical trial was headed by me at the beginning of the 6th centuries. So we call this dendritic cell-based chemoimmunotherapy cancer. Well, we combine use of the dendritic cell vaccination, autologous dendritic cell vaccination, and also conventional chemotherapy in a low dosage. You can see here, we can isolate the mononuclear cell, monocytes, prepare the autologous dendritic cell, and then we let the patient receive the conventional chemotherapy for 5 days. Then we have 2 rounds of the dendritic cell vaccination. So from here you can see, if the patients just receive the chemotherapy, they have the limited objective response. However, if we use the combat therapy of the chemoimmunotherapy, the objective response increased 1-fold. This dendritic cell, we prepared by pulsing them with the heat-stressed auto-tumor cell lysis. So this is just showing you the heat shock protein or heat shock treatment can increase the tumor genicity of the tumor lysis or tumor cells, then increase the capacity of the dendritic cell to activate anti-tumor immune response, both basically and clinically. And also, we identified what's happened to the patients after vaccination. You can see here, there is an increased antigen-specific CTL response, especially gamma interferon secreting T cell. And also, there is a dramatic reduction of the immune suppressive cytokines in the blood of the cancer patient after receiving dendritic cell chemoimmunotherapy. For example, there's decreased presence of IL-10 and TGL-beta in the blood. And also we are wondering what's the biomarkers can select or can discriminate the part of the patients which are responsive to the dendritic cell-based chemoimmunotherapy. We selected dozens of the biomarkers to stain the tissue samples. Finally, we found, which is went back to a study. So there is the increased expression of HSP70 positive tumor cells, and also there are more infiltration of the CD45 RO T cell in the tumor microenvironment. These patients will be responsive to the dendritic cell immunotherapy. So this is give us another functional clues. There is a potent immune suppression at the later stage of cancer patients. So we analyzed several kinds of immune suppressive cells in tumor-bearing mouse in the cancer patients. We identified several kinds of the regulatory cells, including regulatory dendritic cells. This is how we identify this population of the regulatory dendritic cell. As we know, the mature dendritic cell can present antigen to T cells and activate a T cell response. Then one question arises, what's the fate of the dendritic cell after their antigen-presenting and T cell activation? So it's generally accepted they will go to death because they are the terminally differentiated cell. However, when we co-culture the mature dendritic cell with the stroma, especially splenic stroma, we find the mature dendritic cells can go further proliferate and differentiate to another type of the dendritic cells with regulatory function. It can feedback inhibit T cell proliferation initiated by mature dendritic cell by producing large amount of the nitric oxide. We identified this regulation, this population of regulatory dendritic cell in the tumor tissue. So this is the basic study of the dendritic cell in tumor and immunology. So what we learned from the clinical and basic study of dendritic cell-based cancer therapy, so we should further improve the efficacy of the dendritic cell vaccination. How to do it? And we should identify the biomarkers to predicate the patients who are responsive to the therapy. and also suppressing the suppressor, just blocking the immune suppression or inflammation will benefit the cancer patients. So this inspires us to do further research in several aspects. So we should identify what's the innate signals in immunity, which can be utilized to design the new type of adjuvant for cancer immunotherapy. And also, what kind of the immune suppressive substance or molecule in tumor microenvironment and even in the secondary lymph organs of tumor-bearing host, which can be deleted to benefit the cancer patients. And so we try to identify the immune suppressive or inflammatory circulating factors in cancer, which can also be used as the biomarker or the target for cancer immunotherapy. So this is the background. We move to the innate response inflammation in cancer. There are several pathways for the innate response. The toll-like signaling for bacterial infection and the RIG-I for RNA viral infection. So we identified several kinds of molecule on the cell surface or intracellular which can positive or negative regulate the PR signaling in the macrophage or dendritic cell through different mechanisms. For example, the Siglec-G, actually we cloned independently the Siglec-10, the human homologue of the Siglec-G from the human dendritic cell. We found this molecule on the surface can be upregulated by the viral infection. Then it can recruit SCP2 and the SIBO to promote the degradation of RIG-I, then disrupt the TIP1 interferon production, so preventing the overactivation of the immune response, inflammation. And also we figure out one long-coding RNA we called LSM3B. I have several slides to show you later. So we also moved our attention to the nucleus of the immune cell, of the macrophage or the dendritic cell. So we identified several epigenetic modifiers which play an important role in the control of the cytokine production. For example, the TAT2. As we know, the cytokine production must be initiated timely, but if it continues to present for a longer time, it will induce the harmful immune pathology. So one question to us, how to timely terminate the pro-inflammatory cytokine production at the later stage of innate response, especially the immune system has already eliminated the evading pathogens. For example, negative control of the TIP1 interferon and how to terminate the production of IL-6. Both of them are pro-inflammatory cytokines. So we analyze the gene expression profile of the macrophage or dendritic cells in response to viral infection. Just to take this example, we analyzed the VSV-infected, the macrophage expression profiles. We found there are so many molecules upregulated by the viral infection. And among them, there is the TAT2, which can be also upregulated by Tau signaling that you can see here. Then we analyze the TAT2 deficient macrophages and the dendritic cells. What's their response? You can see when we started to analyze the IL-6 production as early stage of innate response, 4 hours, 6 hours, there was no dramatic changes. However, when we extended the analyzed time course until to 12 hours, there was dramatic difference of the IL-6 production. Especially TAT2 deficiency will enhance the IL-6 production at the later stage of inflammation or innate response. So this reminds us that TAT2 may be the negative regulator of the IL-6 production at the later stage of immune response and inflammation. It's the terminator of the inflammation or help to resolve the inflammation. So this is just one case for us. Now we understand the immune system can interact with the environmental factors and also can be modulated by the intrinsic factors, including metabolic molecules and also epigenetic modifiers. So through this way, we can understand how the immune system is balanced by interaction with the environmental factors. So through this way, we can enhance the enhancer and suppress the suppressor to be helpful to design of cancer immunotherapy. We can also regulate in the regulator to induce the tolerance for the control of the organ rejection. So this is our knowledge about the immune regulation or balanced network of immune system. So what we have done in the last decade, we are focused on the immunobiology of APC and what's their interaction with the cancer cell and what's the inflammation initiated by them in cancer progression and cancer immunotherapy. So we ask the question, what's the role of the innate sensors in host and the cancer interactions, especially at the level of the stromal and the APC and the molecule levels. So I want to share two stories about the two molecules, RIG-I and the Toll-like receptor 3. Both of them are the RNA innate sensor. How about the role of the RIG-I in the immune response? So there are so many post-translational modifications of the innate sensors. You can see this is the Toll-like receptor signaling responsible for the production of the pro-inflammatory cytokines, TNF-alpha and IL-6. This is the Toll-like 3 and RIG-I and also STING pathways for the activation of the IF3 leading to the production of type-1 interferon. So we identified several molecules. Just now I mentioned G, and this year we identified IF-122, also responsible for the post-translational modification of the IL-1. So now the epigenetic regulators are very hot field. So we did the dynamic analysis of RIG-I initiated type-1 interferon production, and then move to how we can identify epigenetic modifier in the control of the RIG-I initiated inflammatory response. So when we analyze the response, innate response, you can see there is the repeat production or the expression of the type-1 interferon, including alpha and beta. And also, accordingly, there is the increased phosphorylation of the IF3. However, when you analyze the level of the RIG-I protein, you can see it's relatively low at the beginning of the innate response, but it exerts efficient role to activate the production of the type-1 interferon. This is the shutdown after 8 hours after viral infection. However, you can see there is a continuous increase of the RIG-I protein even 24 hours after viral infection. So what's this association of the increase or the decreased expression of the type-1 interferon, but the increased expression of the RIG-I protein? There must be some molecule which can terminate the recognition of the pathogenic RNA by the RIG-I, so they can not initiate the phosphorylation of the IF3. So we did the CLIP-seq. We find there are many kind of long-coding RNA which could be bound to the RIG-I. And actually, RIG-I is the RNA binding protein. So we figure out the one molecule, and long-coding RNA SF3 is combined to especially there is no binding of the 3 with the RIG-I before the viral infection. There is a dramatic binding between them after viral infection. So we made the knockout mouse you can see here. This deficient mouse are resistant to RNA-viral challenge with increased survival, and also there was a dramatic production of T4 interferon in their blood, and also accordingly there is less tissue damage in the deficient mouse. And make the story short, we found this molecule can direct bind to the CTD of the RIG-I. So at the later stage of the innate response, there is upregulation of the long-non-coding RNA 3b, which bind to the RIG-I, which competitive with the pathogenic RNA to terminate the recombination of the RIG-I to the pathogenic RNA. Then it terminates the initiation of the phosphorylation of the IF3 by RIG-I. So this is just one mechanism of the immune system. The self-RNA can timely terminate the innate inflammatory response at the later stage by disrupting the function of the RNA sensor. So this be helpful to understand. Self will help ourselves to maintaining the metastasis of the immune system. So this is the ongoing basic research in my laboratory, trying to identify the long-non-coding RNA and RNA-binding protein in the inflammation, immune suppression, and cancer immunotherapy. And we just identified several RNA-binding long-non-coding RNA. And also, I have no time to present the long-non-coding RNA we identified in the dendritic cell. We call this link DC, which was the story in Science 2 years ago. So for the role of the RIG-I in the cancer immunotherapy, so this is the translational study of this molecule in cancer immunotherapy. So as you know, the hepatocellular carcinoma is a serious problem in China with high incidence and mortality, and alpha interferon adjuvant therapy only benefit limited parts of the liver cancer patients. So we collected the tissue samples of the normal liver and fibrotic liver, hepatitis liver, hepatocellular carcinoma, and also the hepatocellular carcinoma tissue from the patients receiving the alpha interferon therapy. Then we did the microRNA to identify this microRNA 199 is a good marker to predict the prognosis of the liver cancer. This is the cancer story paper 4 years ago. And we also are very interested in how we can figure out the patients who are responsive to alpha interferon therapy. So we identified the RSG expression profile after the patient in the hepatocellular carcinoma treated by the alpha interferon. So you can see here. And we collected the tissue sample from the liver cancer patient, and then also did the microarray analysis. You can see here there are several RSG downregulated. Among them, one molecule attracts our attention. It's RIG-I. It's the RNA sensor. So we are asking a question, what's the role of the RIG-I in liver cancer prognosis, and especially in the alpha interferon therapy of the patients? So we detected the RIG-I expression in the liver tissue, and we also confirmed this. There is a decreased expression of RIG-I in the liver cancer tissue through in different cohorts. And also, we analyzed the expression of the RIG-I in the liver cancer tissue with the survival of the patient. You can see, with high expression of RIG-I in the liver cancer tissue, the patient will have the prolonged overall survival. We have two independent cohorts to show this. And also, how about alpha interferon therapy? And you can see here, if you have overall survival of the cancer patient after receiving alpha interferon, there are almost no dramatic changes. However, when you distinguish the patients into two groups, one has a high expression of the RIG-I in the liver cancer tissue. These patients will have the prolonged survival after alpha interferon therapy. If the patient has a lower suppression of the RIG-I in their tissue, they have no response to the alpha interferon therapy. So the RIG-I expression level may be the good marker to predicate the responsiveness of the alpha interferon in this part of the cancer patients. Finally, we figure out the mechanism behind. The RIG-I can bind to the STAT1, then preventing the dephosphorylation of the STAT1 by phosphatase SHP1. Through this way, it can maintain the phosphorylation status and promotes the nuclear translocation of the STAT1 in response to alpha interferon stimulation. So the RIG-I, the RNA sensor, can act as the tumor suppressor and also can be utilized as the biomarker of the alpha interferon therapy, at least in the liver cancer patients. And also, we went further to analyze why the liver cancer, the low expression of the RIG-I in liver cancer. And we try to provide at least partially, the mechanistic explanation why most of the liver cancer patients are men, not women. So we detected that, found there is high expression of RIG-I in normal liver of women population, and very low in the men. So this is just provides the process. But at the same time, it also indicates why the women population have increased instance of autoimmune disease, such as lupus, with increased production of the type 1 interferon. Then another story about the TLR-3 in the cancer metastasis. So now we have so many reports about the role of the TLR expression on the tumor cell. Actually, 10 years ago, we also have 2 papers showing the tumor cell can express TLR4, and the TLR4 ligands can increase the drug resistance, the death resistance of the cancer cell, and also promotes the invasive capacity of the cancer cell. So now we want to address what's the role of host TLR receptors in the tumor metastasis, especially in pre-metastatic niche in the distant organ. So we ask this question by challenging the TLR receptor deficient mouse by inoculating the Lewis cancer cell and the B6 melanoma subcutaneously, and observe the spontaneous lung metastasis by removing the primary tumor. So we check this. As you can see here, there is a less pulmonary metastasis in the TLR3 deficient mouse. And also there is another interesting observation. There is a less pulmonary metastasis in the type 1 interferon receptor mouse. That said, we are now performing experiments to confirm this. Now we show the experiments about the TLR3 and the lung metastasis. So you can see here, there is dramatic decrease of the lung metastasis in the TLR3 deficient mouse, but not in the TLR4 and TLR9. And this is the significant decrease of the lung metastasis the members. And you can see here, TLR3 knocked mouse have the longer survival after tumor inoculation. And importantly, the pre-metastasis niche is the new concept for cancer metastasis. It's always 10 years ago proposed. The primary tumor can indicate the distant organ for creating the favorable environment for the future metastasis by releasing the factors in the blood. So there are so many markers for the pre-metastatic niche, including S500, MP9, or BV8. So we detect the expression of the pre-metastatic niche markers in the lung, especially in the toll-like receptor deficient mouse in response to tumor inoculation. You can see here, there is a dramatic decrease of the pre-metastatic niche markers in the TLR-3 deficient mouse, and also there is a decreased expression of the fibronectin in this. So what's the mechanism responsible for this? So make the story short, we analyze different types of the immune cells. Finally, we find the neutrophils involved in this. You can see here, after the inoculation of tumor cells, there is increased recruitment of the neutrophil to the lung. However, without toll-like 3, there is less neutrophil recruited to the lung. Then we also find there is the decrease of the neutrophil recruited to the spleen or in the presence of in the peripheral blood, but not in the bone marrow. So we detected the neutrophil attracting chemokines. You can see here there is a lower level of chemokine in the serum and also in the buff of the toll-like receptor deficient mouse in response to tumor challenge, you can see. So the toll-like 3 deficiency prevents lung pre-metastatic niche formation and spontaneous lung metastasis by reducing neutrophil recruitment to the lung. So next we want to know what's the mechanism for the decreased expression of the chemokine production and the impaired neutrophil recruitment. By performing the bone marrow transplantation, we confirm the toll-like 3 expression in the stroma is responsible for this phenomenon we observed. At first, we are very interested in the toll-like 3 expression on the myeloid cell. Finally, we saw it was not. So the stroma cell in the lung will be responsible for the neutrophil recruitment. Then we analyze the expression of the toll-like 3, different type of the immune cells in the lung. Finally, we found it's not the immune cell. It's the highly expression of the toll-like 3 in the lungs epithelial cell. And we finally figure out it's the alveolar type 2 epithelial cell expresses high level of toll-like 3. It's here. And then we rescue the toll-like 3 expression in the lung, especially in the toll-like 3 deficient mouse. And we find here, you can see, it can rescue the neutrophil recruitment and the lung metastasis. You can see here, and by the toll-like 3 recruitment. And what is this possible for the neutrophil response for this? When we delete neutrophil and also reduce the lung metastasis, it's that to say toll-like 3 expression, chemokine production, and the neutrophil recruitment are all responsible for the increased lung metastasis. So alveolar epithelial toll-like 3 expression is critical for the lung metastasis. And then we ask the question, what's the molecule responsible for the toll-like 3 activation? The primary tumor, the lung metastasis, is a long distance. So the primary tumor must release some soluble factors. Then it can indicate the lung microenvironment by these releasing factors. As we know, the exosome derived from the tumor tissues can have this function. They can inhibit immune response. They can also indicate the immune system to be favorable for their escape, especially by inducing the immune suppression. So we isolated the tumor exosome and injected them to the normal mouse. They can see here, this exogenous exosome can be accumulated in the lung and can also be uptaken by the type 2 epithelial cell. And then we can confirm this tumor exosome can upregulate TLR-3 expression in the epithelial cells and also can activate them, leading to the increased production of the chemokine. Without TLR-3, there is less production of chemokine. So that's to say that tumor-derived exosomes can induce the chemokine production by the epithelial cell while TLR-3. Then we also analyze what's the pre-metastatic niche markers expression in the mouse. You can see here increased expression of the fibronectin in the wild-type mouse. However, less expression of fibronectin in TLR-3 deficient mouse after administration of the tumor exosome. So tumor exosome can also upregulate the fibronectin production in the mouse while TLR-3. And then the same phenomenon was observed for the chemokine production and also neutrophil recruitment and lung metastasis. So what's the molecules containing in the tumor exosome responsible for the activation of TLR-3? They must be RNAs. So we treat the tumor exosome with DNAs, RNAs. Only treatment of the exosome with RNAs, it can block the activation of TLR-3 by the exosome. So the exosome RNA are responsible for the upregulation of the TLR-3 expression in the epithelial cells and also responsible for the chemokine production. And we did the RNA-Seq of the tumor exosome RNA. We found there are enriched small nuclear RNA, which are potential ligands of TLR-3. So finally, we translate this basic finding to the clinic, and we find high expression of TLR-3 in the lung cancer correlate the massive neutrophil infiltration. And also, more TLR-3 expression and more infiltration of neutrophil are positively correlated with the increased production of pro-metastatic niche expression. It's called S100, A2, and A7. Importantly, high expression of TLR-3 and more neutrophil infiltration predicate the shortened survival of the liver cancer, of the lung cancer patients. So to make the story, to summarize the story, the primary tumor releases the exosome, which contain RNA, especially the small nuclear RNA. Then it can circulate and move to the lung to upregulate TLR-3 expression and activate TLR-3 in the type 2 epithelial cell, leading to the expression of the chemokine, especially neutrophil, attracting neutrophil. Then recruit more neutrophil to the lung, so forming the pre-metastatic niche, favorable environment for the colonization of the circulating tumor, then promote the cancer metastasis. So we just identified one molecule, TLR-3, in the formation of pre-metastatic niche. So this is so many molecules must be involved in the recruitment of the tumor-promoting immune cells, including the MDSA, regulatory T cells, regulatory B cells, type 2 macrophages, and even the previously unknown population of the human myeloid, bone marrow myeloid cells. So we should pay much attention to identify the more factors which can drive the formation of the pre-metastatic niche, especially in the distant organ with cancer metastasis present. So this is the one question we try to identify. We also summarize the 6 characteristics of the pre-metastatic niche. And 2 of them are the inflammation and the immune suppression. And also there are several molecules involved in the genesis and the vascular permeability, and also involved in the lymphogenesis. However, we should identify the immune suppressive cell and also inflammatory molecule in the process of the forming of the pre-metastatic niche, especially driving the stroma cell with the upregulation of the pre-metastatic molecule. So if we identify this, we can block it and reduce the expression of the pre-metastatic niche molecule. Then, especially together with adaptive immune transfer or checkpoint blockade, must be improve the clinical outcome of the cancer patients. So this is what I want to report to you. Finally, I want to thank the students in my laboratory, especially for Dr. Yanfang Liu and Yan Gu for the TLR-3 story in pre-metastatic niche formation, and Dr. Jing Hu for the RIG-I story in liver cancer. Thank you for attention. I would like to take.
So I'm sure Dr. Xuetao would be happy to answer questions.
Yeah, it's a very nice story. Actually, we know during the infectious, including the viral infectious and bacterial infectious, the toll-like receptor 3 can recognize RNA, but actually it's quite different from the host RNA. But for your study, you showed that toll-like receptor can recognize tumor cell-generated RNA. So because the tumor cell is generated from our host cells, so what's the difference of the RNA recognized by the toll-like-3 with the bacteria or virus RNA?
A good question. Actually, we did not find the signature of the tumor-derived RNA responsible for toll-like-3 recognition and activation. We tried to do this, but unfortunately, we did not have that data. However, there are so many mutants in the tumor cells. They must be different with the normal cell. So we have the RNA-Seq data. We find the RNA fractions are enriched of the small nuclear RNA, totally different from the expression profile from the normal epithelial cell. So now we have no advanced tool to figure out what kind of the pathogenic RNA generated by the tumor cell. And I have to mention that the RNA sensor can recognize the self-RNA normally. For example, the RIG-I just showed you the story. It can bind to the long-coding RNA SLM3B. We find RIG-I can also bind to the 28th ribosome RNA, but very transient binding and repeatedly released. There is no enough affinity to activate the ATP's activity of RIG-I and release and also release the auto inhibitory loop. So I think binding affinity of the pathogenic RNA or RNA to the sensors will determine the outcome of the molecule in the pathogenetic process or the physiological process.
Okay, thank you.
Xuetao, over here, very beautiful story. So, as you know, many things are double-edged swords, and one of them, I guess, could be TLR-3. So, poly-IC, as you know, is a very good adjuvant, and acting through TLR-3 to activate dendritic cells and antigen presentation and so forth. But you also described how it's involved in recruiting neutrophils that are immunosuppressive. So I'm wondering how those two interface and whether it might be possible, for example, to improve the adjuvanticity of something like poly-IC to induce TLR-3 by blocking the chemokines that recruit neutrophils. So now you have the beneficial effect without the adverse effect.
Thank you, Jeff, for the very important question. So actually, this is the two phases of the innate molecule in tumor progression and cancer immunotherapy. So there are also two phases of the neutrophil in cancer. In the acute phase, the neutrophil can kill the tumor cell, especially activated by the adjuvant molecule. However, naturally, especially in the chronic inflammatory process, the neutrophil, maybe they have a different subsets of the neutrophil secreting several kinds of the pro-inflammatory cytokines, maybe. So the natural arising neutrophil can promote cancer metastasis, which have been confirmed by several laboratories. So poly-IC, after ejected in vivo, especially in the tumor tissue, it can activate the acute inflammatory response, which also so many innate immune cells can work together to kill the tumor cell. However, in the natural process of the tumor metastasis, the primary tumor can indicate the stromal cell, not the inner tumor, the stromal cell, to be favorable for their colonization. So this is depend on the strength of one molecule acting and the size of the one molecule exert their function, and also the stage of one molecule they would like to play.
Thank you.
Thank you. This topic is very close to my heart, too close for comfort. I've been in inflammation research for the last 35 years. I came to this institution and struggled heavily to promote the role of inflammation in cancer, which was heavily opposed, rejected, downplayed, and now I see everywhere that inflammation has something to do with it. And another comment and then a question is that the yin and yang of inflammation that you mentioned was introduced to the Nature several journals in 2006 and 2007, and then I finally published it in 2008. With regard to the eosinophil, the dendritic cells and macrophages you mentioned, if you are genetically modifying the macrophages, have you noticed any compensatory mechanism that other innate immune system gets involved? Because this is what we have seen, the interaction and synergies between the host immune cell, which is mast cell, and then interaction with activation of eosinophil infiltration and then macrophage infiltration that leads to the first evidence for a direct link between inflammation and tumor development, which I paid heavily for promoting, even through the legal channels.
Yeah. Thank you for the good comments. And as to the role of the macrophage subsets in cancer, immune response or the immune escape, there are controversial reports. And I think this is a good topic to be further investigated.
Yes.
The inflammatory adjuvant can polarize the type 2 macrophage generation, and especially in the tumor microenvironments, which can feedback promote the cancer metastasis and also render the tumor cell more resistant to the death reduction. However, some researchers tried efforts to polarize the TIP1 macrophage generation in the tumor microenvironments. So trying to utilize the TIP1 cytokines to activate their function. I think they figure out the several molecules involved in this. I cannot remember the exact name, but their approach to change the tumor promoting status of macrophage to the tumor killing status of macrophage in the tumor environment.
M1, M2, right?
Yeah, just one case about the dendritic cell in the tumor environment. As it's very established, the dendritic cell in the tumor microenvironment are all the tolerogenic, all the regulators. Mm-hmm. However, 2 years ago, there are several reports about the CD103 positive dendritic cell, which can initiate T cell response, even in the environment of the tumor tissue. Yes. So there must be some tumor killing immune subsets, including macrophage and dendritic cells, and even in the tumor microenvironment, which need us to identify further.
Yes, thank you.
One last question.
Okay, my name is Pepe Zhang, and I'm in the NIDCR, and I'm the newcomer in the inflammation scope, so it's my great opportunity to get the instruction. My question is whether you have any comments on the epithelium-mesenchymal transition with the interaction with the inflammation, because EMT is also a good model for the cancer. So I'm thinking about this.
So I'm not very familiar with EMT so in this field. I think there are two kinds of the cells, resident immune cells and the migratory immune cells, which can interact with the stroma in the distant organ. I think epithelial cells, endothelial cells, and also other kind of cells, stroma cells, must be interactive to the immune cells. Not only to the cell-to-cell direct contact, but also can interact by the soluble factors secreted by two kinds of cells. I think there must be the inflammatory response to the factors released by the immune cells, especially immune suppressive cell, in the process of tumor genesis or tumor metastasis.
Thank you.
So let's thank our speaker for an outstanding presentation.
Thank you.
There will be a reception in the library, so if you have additional questions, I'm sure Dr. Xuetao will be happy to answer them.
Thank you again.
Thank you again.